I'm going to ask two questions, both of which are interestingly incorporated into that final paragraph of Tom Webb's review. And they're questions motivated by deep time. And then I will try to illuminate those questions by looking at a single issue that we can track through almost the entirety of the known geologic record. And so the questions are, without question, there are limits on the number of redox reactions that are available for metabolism. There are only so many ways of being an autotroph or a heterotroph. So there have to be important points of comparisons between ecosystems in time and space. But insofar as the ecosystems represented in the geologic record consist almost entirely of species that are distinct from those of modern ecosystems, one really does want to ask the question whether species matter when we try to make generalizations about ecosystem structure and function. And then there's the physical aspect of ecosystems. Does the changing nature of resource availability through time influence ecosystem structure and function? We know that's the case in space. So what happens when we look in deep time? And the system I'm going to explore is simply to look at the geologic history of marine phytoplankton. So let me just give a very slight overview for a field that Chris knows much better than I do. If we look at the nature of primary producers in the oceans today, there's a spatial structure to that, and it is related to resource availability. So if we look at places like mid-ocean gyres where resources are low, that tends to be where cyanobacteria in particular have their greatest proportional representation in the plankton. And as resource availability increases, we have the addition of different eukaryotic groups, and particularly at high resource availabilities, we tend to see an important contribution from diatoms. And importantly, as we go from low resource availability to high resource availability, not only do we see more primary production, but we see a larger proportional contribution by heterotrophs as well. Now, if we ask at the broadest scale, what does the history of phytoplankton in the oceans look like? It actually bears some comparison to what we just saw in space. We have evidence that Earth had a biological carbon cycle in the oldest rocks we can look at. So sometime between 3.5 or 4 billion years ago, we do start to have photoautotrophs, obviously bacterial, with a very large component by anoxygenic photoautotrophs. Interestingly, we don't see major ecological representation by eukaryotic phytoplankton until Earth history is about 85% over. We start seeing green algae toward the end of the Precambrian. And then if you ask, when did the major chlorophyll A plus C phytoplankton that are so ecologically important in many of today's oceans, the dinoflagellates, the coccolithophorids, and diatoms, they really only begin to diverge after 250 million years ago, first the dinos and coccoliths, and then diatoms. So what I'd like to do is put that history within the framework of what we think we know about resource availability. And I'll start in the Archean, that is the oceans before 2.5 billion years ago. And my point of reference will be that little cartoon that you see in the lower left, which comes from some early work by Philip Froelich. And it basically says something that most of you know, and that is if you ask the source of the phosphate that is available for primary production on continental shelves, some of it comes from runoff from land, and some of it comes from remineralization of organic matter at depth and upwelling of nutrients. In Froelich's cartoon, he suggested it was about 10% runoff and 90% upwelling. There is data since that time that suggests in many places it could be as much as 99-1. And the point I want to make is that for at least the first billion years of primary production on this planet, both of those fluxes were much lower than they are today. So for example, if we look in the Archean world, before 2.5 billion years ago, increasingly evidence suggests is that there was very little continental emergence. Earth was a water planet for its first, say, billion or 2.5 billion years, and that would, of course, severely limit fluxes of phosphate from the weathering of emergent rocks into the oceans. At the same time, for organic matter that is produced in the oceans, are real limitations on oxidant availability to remineralize that organic matter, something that Eva Stuyken first brought to our attention. Not only was there no oxygen available in those oceans, but because of that, there would have been very little sulfate, very little nitrate. So organic remineralization has low efficiency. And to make matters worse, for any phosphate that was released by mineralization in the deep ocean, since those oceans tend to be ferruginous, that is, they had ferrous iron in solution, there would be a very high tendency of that phosphate and ferrous iron to react to form a mineral called vivianite, which has very, very low solubility. So the long and the short of it is that rates of primary production must have been very low. If you look at that right-hand diagram from a paper we published last year, it shows a number of estimates of primary production through the Archean into the earliest Proterozoic. Some of them are based on electron donor availability for a world dominated by anoxygenic photosynthetic bacteria, some for phosphate availability, but they basically all say the same thing, that probably for the first billion or 1.5 billion years of biological history, you would have had rates of primary production in the oceans on the order of maybe a % or so of today's levels. By the beginning of the Proterozoic, that might go up to maybe 10% of today's levels, but it would have stayed there until about 600 million years ago. So when do we first start seeing evidence of eukaryotic participation in the phytoplankton? The best record we have of this actually comes from molecular biomarkers in ancient sediments, and this is from a nice paper from the lab of Jochen Brox in Australia. And what the diagram shows is that if you look at the sterane abundance and composition before this great global ice ages of the late Precambrian, steranes, which are the geologically stable derivatives of sterols, they exist in late Proterozoic rocks, but frankly, many of them appear to be stem group steranes that are not represented today. And there is no evidence that eukaryotic algae are major producers of those steranes. You go into the early Ediacaran period, beginning a little less than 635 million years ago, and we have one place where stigmasterane, which is derived from green algae, is important, and a couple places where it's a trace constituent. And then by 600 million years ago, basically everywhere you see molecular evidence of phytoplankton in the rock record, there is an ecologically important component of green algae, and that goes into the Cambrian. And that's interesting because we have evidence that eukaryotes and eukaryotic photoautotrophs existed a long time before that. If you look at that rogues gallery in the upper part, there are cenocytic green algae at 1 billion years ago. There are Bangiophyte-like red algae 1 billion years ago. And in fact, there are eukaryotes of unknown affinities, quite possibly stem eukaryotes that go back about 1,800 million years ago. So why would the phytoplankton change composition around 600 million years ago? And once again, I'm going to blame phosphorus. What you're looking at on the left, particularly on the left-hand side at the top, is something called the Doshantou Formation in China, a little less than 600 million years old. And that cliff that you're looking at consists entirely of resource-grade phosphorite. In fact, there is more phosphorite, which is basically almost completely made up of phosphate minerals. There's more phosphate in the Doshantou Formation than there is in all known formations from all continents from all of previous Earth history. So there's a state change in the availability of phosphorite beginning at this time. And we might ask, why would there be this state change, which clearly has some biological repercussions? We could ask, well, maybe erosional fluxes increased during the Ediacaran period. And the way to go about that, and I won't go into it in detail, but I'd be happy to answer questions, is to look at the record of strontium isotopes preserved in carbonate rocks. They are proxies for seawater strontium, which in turn reflects the relative fluxes of hydrothermal systems and continental erosion. Simply put, the higher the numbers, the more important the continental fluxes. And there is a sharp increase at just the time we see this state change. So yes, there is evidence for some increase in resource availability there, but there's issues of magnitude and transience. I think by having mountains and the trans-African mountains that formed at this time in Gondwana are among the largest mountain ranges that Earth has ever seen, but they might be able to, say, increase continental erosion fluxes by twofold, but not by tenfold or anything like that. And there's also a question of transience. Once those mountains essentially erode down, the system would relax to its earlier state. So what we need is some sort of positive feedback that can drive us to another state. And I at least would look for this in sulfate. Turns out if you have more continental exposure, higher rates of erosion, you will oxidize pyrite in exposed rocks, drive more sulfate into the oceans. And again, there is record for an Ediacaran state change in the abundance of sulfate deposits on this planet. And that would influence regeneration in two ways. One is enhanced oxidants will increase the efficiency of mineralization. And what you see in that little rainbow diagram on the left from a paper we published last year is sort of the expected amount of phosphate available for upwelling. And you can see that when organic carbon burial efficiency is high, it's reciprocal of remineralization efficiency, then, well, that's associated with, on this Lake Metano example from the real world, that you actually have, if you have anoxic bottom waters and low oxidant availability, you get very little remineralization. Whereas if you look on the left where it says Black Sea, bottom waters are equally anoxic, but they're rich in sulfate, and then you have high rates of remineralization. So we think that this might have started a feedback of, more oxidants, more remineralization, more primary productivity means more oxidants, and you could drive yourself to a new state. The other thing that would be important is as the sulfate inventory increased in the oceans, then sulfate reduction in sediments would actually titrate out that ferrous iron and essentially remove that vivinite sink for phosphate. So the long and the short of it is that we think that something much more similar to a modern phosphorus cycle really takes shape at about the time we start seeing eukaryotes as important ecological components of the phytoplankton. So when do we see the things that we all recognize from modern oceans as primary producers? Again, if you look at coccolithophorids on the left, we first start seeing them about 230 or 240 million years ago. They radiate during the Jurassic and rise to ecological prominence. Pretty much the same thing is true of photosynthetic dinoflagellates. Diatoms start to come in later, but they have a long-term radiation over the last 100 million years to bring them to their current importance. Interestingly, you can argue that there might be biases inherent to that microfossil record, but if you look at the, again, the molecular biomarker record, and this is basically from a database of 10,000 petroleum samples in Phanerozoic rocks, you can see that if you look at C28 to C29 sterain ratios, they are low through the Paleozoic, which means that you're mostly looking at green algae, but on the same time scale as fossils document the radiation of these chlorophyll A plus C groups, biomarkers do the same thing. So it appears to be a real signal. And for the last time today, I will argue that there may be a nutrient flux issue here. One of the things that happens on the timescale of this chlorophyll A plus C group radiation is that we have a remarkable increase in the dynamic topography of the ocean. So this is when you're forming the Rockies and the Andes in the New World. It's when you're starting to form that great mountain chain that runs from the Pyrenees through the Alps on through the Caucasus and the Himalaya. That again is documented by a major increase in the strontium isotope record. And that suggests that once again, nutrient availability is reaching something of a historic high. It's also been suggested fairly recently that the final real oxygenation of deep waters only happens at the beginning of this event as well. So if that's the story, one can ask what the ecosystem consequences of these episodic increases in fee availability would be. And we know that to a first approximation, at least those first two events that I talked about are associated with essentially step changes in the availability of oxygen in the atmosphere and surface oceans. None before the so-called Great Oxygenation event at 2.4 billion years ago, probably oxygen levels around 1% or so of today's levels until around 600 million years ago, and then we begin the final ascent to modern values. That in turn would have important biological consequences. There would be a tremendous increase in the oxidant pools, not only oxygen, but sulfate and nitrate. That would have major consequences for respiration and chemosynthesis. And I think that's important. I must say, just because of my background, I tend to view plants and animals as icing on the sort of ecosystem cake, but I think microbes are the cake. And I think a very important structure for ecosystem function is set in place when we have this full range of redox reactions available for microbial metabolism. Also, as oxygen begins to rise in the atmosphere and surface ocean, cyanobacteria essentially are major ecosystem engineers. They remove alternative electron donors from most surface waters, and therefore they ensure their dominance from then on. The important thing that happens 600 million years ago, or a little bit after, is we start seeing macroscopic animals, and that really introduces the biological and ecological world that we know in the Phanerozoic. And I think you could at least make a case that it is not a coincidence that we start seeing large animals when we do, but that might reflect increases in both food and oxygen. And then finally, it's really interesting that back in the 1970s, a person named Gary Vermeer argued that there was a major revolution in the nature of marine ecosystems beginning in the middle of the Mesozoic era and accelerating to today. And basically, he argued that there was a major increase in predators, in top carnivores. That you can see qualitatively in that you start seeing things like these little noticid gastropod borings in shells. You see a radiation of predatory gastropods in decapod crustaceans with large crushing pincers, of fish that are capable of shell crushing. And that has actually been quantified by looking at damage on shells by Huntley and Kowaleski, and you can see that the evidence for predation really ratchets up in the later Mesozoic and Cenozoic. We see that in the nature of prey items. If you look at fossils in outcrops from, say, the early Jurassic period, there are bivalves everywhere, and almost all of them are oysters sitting out on the surface. From maybe the late Jurassic through to today, increasingly, the proportionality is such that most bivalves are infaunal. And if you ask why would a filter feeding organism live inside the sediments, predator avoidance is an important part of it. The same thing is true on echinoderms. You see also major increases in the armature and mechanical strength of gastropod species. And just to finish off, if we look at energetics, there's a nice paper by Seth Finnegan et al. Where they show that just based on what we know about metabolic requirements of living gastropods, that the sort of per organism energy requirement of gastropods in the Triassic and early Jurassic is low relative to what it is later. The proportion of gastropods that are predatory, which have higher energy requirements, goes up. And on the right is just a little diagram from a paper that Mick Follows and I wrote a few years ago, which really is based on work done by Mick and other phytoplankton ecologists over many years, showing that at low rates of primary production or resource availability, we expect to have dominance by small plankton, which is that lowermost horizontal line. But as resource availability increases, we tend to have the influx of larger plankton as resource availability increases. And that has the consequence of being able to support more heterotrophs, presumably more top carnivores. So that may link all of these observations. So just to finish up then, over the long arc of Earth history, we see correlated changes through time in nutrient availability, the identity of primary producers, the redox state of the oceans, and ecosystem complexity. And the questions that I leave you with, and I genuinely look forward to hearing your opinions, is, we do have rules of ecosystem function that have been hammered out by ecologists over the years. And to what extent can we take those and apply them profitably to what we see through the long arc of Earth history? Or do we need that arc in order to think about real generality? And I would say, although I spent my time talking about marine phytoplankton, I could have told the same story using terrestrial primary production. One can make a very good case that the world changes when angiosperms become ecologically important. There was a paper in Science magazine 2 weeks ago arguing that until dinosaurs went extinct, they ensured an open spatial structure of forests that we seldom see today. So I will then leave you just with the question of how well can we take models of ecosystem function that we see and apply to variability in space and apply them to variability in time. So thanks, Chris. So Chris is gone. There he is. So you went to sleep already?
Do you hear me?
Yes.
Okay. May I ask a question?
Yes,
sure.
Please. Yeah, I'm not sure if I can't see people's hands raised, so just shout. That's fine.
Okay.
Yeah.
So for what I understood, even though there are bacteria since very old time, they were not widespread in the ocean. Did I understood well? They started to accumulate in the ocean only recently due to increase of resources. This is what your hypothesis, your analysis?
Yeah, I think that's right. I think for at least half of Earth history, bacteria and archaea were the only constituents of life in the oceans, but that their abundance would have been limited by resources, and their metabolic variability would have been limited by low oxygen.
And if I could continue on this, so the myth that the paradigm that life originated in the ocean, sometimes we heard this, probably is that originated in aquatic environments, but not necessarily in the ocean. Can we say
I think that's correct, that more and more thinking from people who do origin of life research looks at things like, if you have prebiotic chemistry in the oceans, you have a tremendous problem of dilution. Whereas if you're looking at freshwater environments, you can actually concentrate those intermediates that are important in biogenesis. And that doesn't mean that you have to throw away the ideas of hydrothermal systems. As I keep telling my origin of life friends, there is a place where you can go to with a few hours flight where you can see hydrothermal systems on land feeding lakes, and that's Iceland. So I think that those kind of environments may have been key, which then carries the suggestion that life in the ocean then got there by colonization and not necessarily by origination.
Oh, okay. Thank you. This is so. Ocean has been colonized quite recently. Thank you.
Does anybody else want to shout up? I can also ask in the meantime, Andy. Was the rise in abundance of eukaryotic autotrophs, in your opinion, responsible for the snowball Earths then that followed the increase in the ocean in drawing down CO2?
Yes, we explored that about a decade ago. I think most of the rise of eukaryotic phytoplankton postdates those glaciations. And just to back up for those of you who don't follow the geologic literature, between about 714 million years ago and 635 million years ago were two of the largest ice ages our planet has ever seen. You have evidence of glacial ice at the equator at sea level, which is a little bit different from anything that we've experienced in the last few million years. Clearly, that reflects a big drop in CO2, which I think opinion now might suggest that that was more tectonically driven. But I think there is a biological part of it, and that is today it may be pretty much impossible to have this runaway glaciation that goes to low latitudes because of the way that vegetation has changed the albedo of the continents. In the absence of vegetation, we have at least 3 times in Earth history that we had this global glaciation. Once you get land plants, you never see it again.
Thanks. Any more comments from the floor? Yes, Sergio?
I would like to ask you one small question regarding the phosphorus limitation. All the through in history you presented today, we're always talking about phosphorus limitation of the ocean, while today many people assume it's a nitrogen limited ocean. So would you please comment on that? It has been a change in the why exactly what happened to that?
Yeah, no, that's a very good question, and nitrogen is very important. Very clearly, there is at least regional nitrate or nitrogen limitation in the oceans today. I think at the levels of phosphate that we're talking about in early Earth history, it's not clear that Earth would have actually needed nitrogen fixation, because abiotic nitrogen fixation from lightning can probably provide all the nitrogen you would need. And we do have evidence that in Proterozoic oceans, we have an active nitrogen cycle that includes bacterial nitrogen fixation. So I guess my view is that on the kind of timescales that I'm talking about, nitrogen fixation will help to maintain something of a balance between phosphate availability and primary production. But I certainly agree with you that at least in some times in some places in today's oceans, and presumably at some times in some places in oceans over the last 600 million years, nitrate, or as we know in the modern oceans, iron can actually limit primary production. So we want to think holistically. In 20 minutes, I just picked one and went with it.
Excellent. Perhaps in the interest of time, then we better move on. Thanks. Great opening, Andy, for the meeting. So, Hélène, I'm not sure if you are there, but I presume you are there. Yes.
Okay.
Nice to see you.
Yeah, hi.
So, Hélène Morlon is colleague of mine at the École Normale Supérieure in Paris, and she's going to talk about a similar topic, but with very different approaches. So, over to you, Hélène. Thank you.
Okay. Thank you, Chris, for organizing this, and thank you. You gave me the opportunity to think again about things that I haven't been thinking about for a while, and in particular, the relationship between biodiversity and ecosystem function. So I actually work on the dynamics of biodiversity in the deep past from a neontological phylogenetic perspective. And in this field, we don't think so much about the relationship between biodiversity and ecosystem function. We focus a lot on the dynamics of biodiversity. So what I decided to do today then was to talk first about why it could be interesting and important to study the relationship between biodiversity and ecosystem function in the deep past, how we can go about that, how we can study this relationship in the deep past from a paleo perspective, and then how we might study it using a phylogenetic perspective, which is not really done much. Okay, so obviously, like characterizing the relationship between biodiversity and ecosystem functioning is a priority in the context of the current biodiversity loss. So in the context of biodiversity erosion, where we are losing species richness and we're losing functional diversity, and we don't really have a super good idea on how this feedbacks on ecosystem function and in the end, ecosystem services that we benefit from. So this is a super big field of research, for which we have people who have funded this field here, notably Michel Laureau was here. And in the main ways that this relationship has been characterized and studied is on one hand, from a macroecological perspective, looking at spatial patterns of biodiversity, where we have been able to look at relationship between productivity and diversity, and thinking more that diversity, species richness might be limited by the amount of productivity or resources that we have available in ecosystems. But otherwise, it's been studied mostly using theoretical models and experiments on rather short time scales. I mean, there's pretty impressive experiments that have been done over decades, but it remains it remains kind of small scale experiments that have helped a lot in characterizing this relationship between diversity and ecosystem function. However, or I don't know if it's that recent, but there's been recognition that there might be a discrepancy between these experiments on short time scales that are done and what might happen in natural systems in the natural world. So here it's from the introduction of a special issue that was published in P2RSB in 2016 on the subject, and saying, so yeah, what is the urgent question that warrants this special issue, this discrepancy between types of experiment done and the natural systems undergoing biodiversity change. So the experiments are carried under controlled, standardized conditions on small scale plots over short periods of time. And yeah, they have helped a lot in unravel many mechanisms linking biodiversity to ecosystem function, but they might have a limited capacity to predict these relationships in multi-trophic communities and at larger spatial and longer temporal scales that are relevant for ecosystem services to human societies. Recently, there has been some also like efforts to scale up these types of research. And for example, here we have an example of a study published a couple years ago in Switzerland, where productivity was censused over 16 years in a 60-day period using satellite-based proxies for productivity. And in this case, so biodiversity was censused twice for plants, birds, and butterflies, and then productivity at a pretty fine timescale resolution. And demonstrating here a relationship between species richness and both productivity and the stability of ecosystems. And here again, so it's really now like really going in the natural world and at big spatial and temporal scales. But again, in terms of diversity, there is still the use of the space for time analogy, where diversity here is censused in a 16 years period, but we don't have big fluctuations in species richness that we can try, temporal fluctuations in species richness that we can then try to correlate with either ecosystem function or ecosystem stability. So and so there's also like this recent review from Andy Gonzalez and Michel and other co-authors who has really called for the scaling up of the biodiversity ecosystem functioning research and proposing a different ways that this can be done. And there's one way that where we have natural experiments of changes in biodiversity through time that might be potentially related to changes in ecosystem function through time, and that is through the past. So and the point has made several times how the past is a natural experiment with many big biodiversity crises that have been going on, mass extinction events, big changes in environmental conditions, and these past natural experiments can help us to understand how biodiversity might respond from environmental crises, and potentially also help us to understand how biodiversity and ecosystem function may respond to environmental changes. So here I put key papers that have been published recently, making this point on how paleobiology or looking at the paleo record can be useful in conservation biology. And in these papers, there's really not much about talking about the biodiversity ecosystem function relationship. And on the other hand, for example, in the recent paper by Andy and Michel, there's not either really talking about how the paleo record can help us look at this relationship. So we were talking about divide in fields between the terrestrial and the marine scientific field. Here again, it seems like there's still a pretty big divide between ecologists that have been working on the biodiversity ecosystem function relationship and paleobiologists who are concerned with the conservation aspect of what their science can bring, but haven't been thinking about the relationship between biodiversity and ecosystem function that much. There have been some studies from the paleo record, but not many, and I've tried to find them. And in particular, in paper by Link, selected key research questions, including this the relationship between biodiversity and ecosystem function, and try to ask whether paleoecology can be a good way to address this question. And so first, like the what we can see is that in the paleo record, biodiversity, when we use microfossils, the focus is more on biodiversity than on productivity. So we look at effects of climate change on levels of species richness rather than really productivity. But there are ways, as Andy has shown in his talk, to look at proxies for productivity or other ways to somehow quantify ecosystem function in the past. So there are ways for some really good fossil groups to look at absolute abundance or to use geochemical proxies. And there's also sometimes where we can have measures of some way to measure ecosystem structure, and so to look at ecosystem stability. So here's an example from, try to remove that, okay. Yeah, so here's an example from a paper by Wolfgang Kissling, who looked at variation in reef species diversity through time, as well as some measures of reef structure, where he looks at ecological Euclidean distance in ecosystem measures in time in successive time bins, and in this way is able to look at how species richness is related to ecological stability. And here he was able to demonstrate a pretty strong relationship between biodiversity and stability. But in this case, he also had a way to look at productivity, and there was no relationship between species richness and productivity. Let's see. Another study here on deep sea ostracod for the last 20,000 years from the North Atlantic Ocean, where productivity was measured looking at proxies for abundance in the ostracod microfossil record. And here, what was demonstrated was a strong relationship between species diversity and productivity, but not with stability. So I'm sure this is not my field, and I'm sure there's other examples, but it doesn't seem that there's that many examples of this relationship being described or characterized in the fossil record. So now, how could we go about studying this relationship using a phylogenetics perspective? So that's more the field that I work in. So in this field, what we're able to do is to use the phylogenies of present-day species, potentially also integrating some fossil species, but mainly extant phylogenies from extant species, and the traits measured of these extant species to understand how diversification has proceeded through time. So to understand the pace at which speciation and extinction occurs through time, and how traits evolve through time. So these approaches are based on stochastic models that we can fit to the data we measure, in particular, so comparative phylogenetic data to infer past dynamics of speciation, extinction, and trait evolution. The relationship between environmental change and diversity that has been studied in this field is more looking at diversification directly rather than biodiversity. So diversification in terms of diversification of species, their speciation, extinction rates, and their rates of trait evolution, rather than directly levels of biodiversity or levels of productivity. And so what people have done, including people in my group, is to develop models where we can directly incorporate measurements of past environmental variables. So here, for example, temperature fluctuations through time in models of diversification and phenotypic evolution to then estimate how these past environmental variations have influenced the diversification of various groups. Here we have examples on vertebrates. Here, on the left, it's on the effect of changes in temperature on speciation rates, and on the right, on rates of trait evolution, where what we see for diversification is for positive effect of increasing temperature on diversification, with speciation rates that tend to decline throughout the Cenozoic as temperature cools down, and rates of phenotypic evolution, here it's body size evolution, that increase throughout the Cenozoic in both bird and animal groups as temperature go down. So with these types of models, something that we could do if we have proxies for productivity in the past would be to look at least to investigate correlations between diversification in the past and productivity. So diversification, productivity relationships. Here is an example where the perspective was more the traditional perspective of looking at how different abiotic or biotic variables changing through geological time have affected the diversification of diatoms. And this is a study we did with Chris using Teradata, which allowed us to build a phylogenetic tree for diatoms, and to then look at what are the different environmental correlates that might influence diversification rates in diatoms. And what we see here is a pretty big support for models where there has been an effect of CO2 concentration, atmospheric CO2 concentration on diversification rates. And here, so that's like this question that we were wondering, what's the cause, what is the effect? And it could be that what we see here is more of an effect of diversification on declines of CO2 concentration during this time period. Okay, so what's done is more to look at diversification than diversity. However, even though, I mean, this gives really a rough estimate, I don't want to make it look like we can estimate diversity through time in the past reliably, but we can have estimates of how diversity changed through time using these techniques. So not only looking at rates of diversification, but how the number of species change through time. Here is an example in the Cetaceans or in Dendroica using different models, or using a recent model that we developed where we use data augmentation that allows to augment an observed phylogenetic tree of extinct species with extinct lineages, according again to a model that we feed to the data, and that allows to reconstruct the number of lineages through time from the molecular phylogeny. So with, if we have proxies for productivity through time, and we use these types of way to reconstruct diversity through time, and we can start to try to look at the relationship between diversity and productivity in the past using these approaches. Okay, so that would be ways that could be thought of to address this question in the deep past. But as I told you, I mean, that I'm aware of, this is not really done in the field. But something else that have been looked at more and makes sense is the fact that, and so not thinking about the relationship between biodiversity and ecosystem function in the past, but the fact that the past history of diversification and phenotypic evolution does influence present-day ecosystem functioning and its response to biodiversity loss. So this is more research that has been done for a while, thinking about whether it's species richness or more phylogenetic diversity or functional diversity that explains ecosystem function, and if obviously like the way that groups diversified and that niches were occupied according to different models of niche evolution is going to impact the relationship between biodiversity and ecosystem function, and what we can expect to see as we lose species in the present day. So here, for example, in this paper by Cadot, we see that under different models of phenotypic evolution, we can have stronger or weaker relationships between species richness and ecosystem function in the present. And we can also have models where, according to how traits evolve, there wouldn't be any relationship between species richness and ecosystem function. And this point also that the past history influence the relationship between biodiversity and ecosystem function, as also been made in this paper, where the authors showed that the diversification in stickleback affect ecosystem function in different ways. So yeah, maybe that's the other way that we can import here, like the types of approaches we have to study how traits evolve to inform the relationship between species richness and ecosystem function. So, I mean, from my perspective, again, that's not exactly what I work on, but it seems like we know little about the relationship between biodiversity and ecosystem function in the deep past, and that it's not easy to have proxies, for example, for primary productivity or ecosystem function or even levels of diversity in the past, but that there is more that could be done using either paleo and phylogenetic approaches to address this question, and that this might be tricky but informative way to scale up biodiversity and ecosystem functioning research. And also just having and not looking in the this relationship in the past, but having an evolutionary perspective to understand this relationship might be important, but this is something that has been addressed more already in the literature. And yeah, this is what I wanted to say. Thank you for listening. Hello?
Yep.
Hi.
Hi. Sorry, it takes me a while to run over. Yes, Maurizio.
Yes, a question. I mean, I see that normally when you want to measure ecosystem functioning, I mean, everybody uses either production or productivity or at most stability. It's not the case to investigate for other metrics, because ecosystem functioning is so generic. All ecosystems function. So how do you rank if it's functioning well or not, besides persistence or maybe, you know, mutation or speciation rate or whatever? I mean, I think that we are missing additional metrics.
I would say that anything we can get that can be a proxy for ecosystem function in the past, I would, would be useful. It seems that maybe primary productivity is one of the easiest or more maybe universal that we can have. For example, if we want to compare, as we were talking, like terrestrial versus marine environments, but I agree, like, I think I don't know, to be honest, I don't know what other metrics of ecosystem functioning we could have in the past. I don't know, but definitely like any type would be useful.
Chris, could I ask a question?
Yes, please.
Thanks, Hélène. That was a lot of fun, and I really like this using phylogenetic studies to look at trait evolution as a measure of ecosystem function. And you set me to thinking about a potential application of that, and that is when we look back over the last 500 million years, there's a number of times of mass extinction, and those have always been studied in terms of just taxonomic diversity. And I'm wondering whether, so for example, at the end of the Permian, you have essentially ecosystem collapse, and whether a phylogenetically based trait approach might help us to better understand both the nature of the recovery and its timing.
Maybe, yeah. I mean, that's the same thing that when I was preparing this talk and, thinking a little bit outside of what I'm used to, but I was thinking that why don't we do that? We don't really do that. So we do use phylogenies to see whether We can't find marks of mass extinction in the tree, but in terms of diversity loss. But in terms of if we see a change in the traits, or we don't do that much, but there are tools there that could maybe be useful to look at these questions.
Yeah, we should talk. That's interesting.
Cool.
Can I also ask a question?
Yes, please.
Bonjour. Yeah, thanks so much. Also from my side. So apologize for my question. I'm more a plot level, small scale, present day BF ecologist. That's why I have not thought that much about these deep temporal scales. And so an issue that we often have when we try to compare results that we get from experiments, the classical BEF experiments with observations, is that there are reciprocal effects between productivity, for example, and diversity, and that there are also many confounding factors like site level environmental conditions. When you have explored this field now, have you also thought about that and how one could address this a bit? Because there might be general differences, ecosystem types that are just more or less productive. Yeah.
No, I have thought, as you have, that yes, it's going to be problematic, and that what we're going to have mostly are correlations, and then, you know, in which direction the correlation go, and all the confounding factors. What we can do, the best we can do, is try to have independent measurements and then control for these confounding factors statistically. But yeah, that's obviously like where, you know, the strength of the experiments you have, where you can control more of these confounding factors that we cannot do. I mean, but yeah, that's the weakness on our side for sure. Yeah.
Can you just go here?
Marina Levy is here to ask a question from the floor.
Hello.
Bonjour Hélène. Hello, hi everyone. Sorry for this maybe basic question, but I'm more of a marine biogeochemist working with present-day oceans, so there are a few notions I'm not familiar with. And one question I have, you talked a lot about ecosystem stability, and I was wondering what kind of indexes, what kind of measures do you use for stability?
So I don't do that, right?
Yes.
But okay, in this case, the one I showed on the reef, the Kissling et al. Paper, they looked at stability by looking at different measures of reef structure, and they looked at the Euclidean distance between the structure in different time bins, in successive time bins.
Okay.
And that gave them a measure of stability.
Okay. So it's really
yeah,
over time. You have to compare different periods of time. That's how they do it. So you need to have data over different periods of time of the same system to really assess stability, which is not an easy thing to do in the open ocean, for instance.
Yeah, but for stability, yes.
Yeah. Thank you.
I don't know, but this, I'm definitely not the expert there, but if there's same like space for time analogies that are done, and so you would look at some measures of variability in space that would give you a measure of stability in time. I don't know, I don't want to go there. There's people who know much better than me here, so they can answer that, how you can measure stability.
Thank
you. Don't want to use temporal data.
Merci.